Welcome back to the deep dive. We're here again to take those complex research papers, the clinical reports, the pathway diagrams, all that dense immunopathology stuff, and really distill it down into practical knowledge for you.
That's the goal.
And today, we're diving into a really critical area. It's at the intersection of neurobiology and immunology, something with, frankly, huge implications for clinical practice.
Yeah.
We're looking at the potential links, the mechanistic links between Autism Spectrum Disorder, or ASD, and autoimmunity.
That's right. And for you, our listener, working in the clinic doing the research, our mission today is really about precision. We want to move past just saying, okay, there's an association between immune issues and ASD.
Right. The correlation is pretty well established now.
Yes, so we're going deeper. We're looking at molecular causation. We're pulling together data that asks how specific triggers think infections, maybe immunological adjuvants, might actually break self-tolerance.
And lead to that chronic neuroinflammation we see.
Exactly, through pathways that are becoming, well, increasingly predictable.
So it's not just correlation, it's about getting down to the how and the why. How does the immune system actually get turned against the body's own tissues in a way that affects brain development?
Precisely.
To help us map this out, we're going to focus on three key concepts that seem to interlock. First up is molecular mimicry, or MM.
Right, molecular mimicry. Think of it as an immunological perfect storm, almost. It's this phenomenon where something foreign, like a piece of a virus or bacteria,
An antigen.
yes, an antigen, shares a similar sequence or even a 3D structure with one of our own self-antigens.
Okay.
So when the immune system gears up to attack that foreign invader, it accidentally also targets our own tissues because they look so similar. Especially, this is key in someone who's genetically susceptible.
Got it. So mistaken identity triggered by infection in the wrong person, basically.
In a nutshell, yes.
Okay, second concept. The effector arm. This is the part that actually does the damage, right? The interleukin-6 and T helper 17 axis, IL-6 and TH17.
Exactly. If mimicry is picking the target, IL-6 and TH17 provide the firepower. The TH17 subset of T cells, along with the cytokines they produce, like IL-17 and IL-22, well, they're notorious.
For driving chronic inflammation.
Yes, they're major players in causing tissue damage across a whole range of autoimmune diseases. So we need to figure out how that initial mimicry event gets amplified into sustained TH17 activity.
Okay, and the third piece, the catalyst.
The catalyst, the external stimuli, things like immunological adjuvants, components added to vaccines to boost the immune response, or environmental factors like an acute infection itself.
These provide the danger signals.
That's the term, yeah. They provide the signals needed to kickstart these really high-stakes inflammatory pathways in the first place. Understanding how these three things, the mimic, the inflammatory pathway, and the trigger, all interact. That's really the key to grasping the whole picture.
All right, let's start unpacking molecular mimicry then. You mentioned it's like mistaken identity. How does this actually play out? It seems like the recent SARS-CoV-2 pandemic gave us a pretty stark example.
It really did. It was almost like a massive, unplanned natural experiment. Immunologists got to watch in real time as a novel pathogen seemed to trigger widespread cross-reactivity.
Leading to autoimmune problems after the infection.
Exactly. The sheer number of reports and how quickly they came out about autoimmune and inflammatory diseases popping up after COVID-19 infection was, well, unprecedented. It suggested the immune system was targeting tissues all over the body.
What kinds of things were clinicians seeing?
Oh, a whole range.
Yeah.
In children, there was that Kawasaki-like multisystem inflammatory syndrome, MISC.
Right.
Then acute neurological problems like Guillain-Barré syndrome, GBS, various forms of myositis, muscle inflammation, IgA vasculitis, sometimes with kidney involvement, nephritis, and even serious kidney diseases like membranous glomerulonephritis, MGN. It was quite a spectrum.
Wow. So the big question then is, how do we prove it's actually mimicry? It can't just be that these things happened after the infection, right? There needs to be a molecular link.
Absolutely. Temporal association isn't enough. You need to pinpoint the specific sequences, the actual molecular structures, where the virus looks virtually identical to human proteins.
And how do researchers even begin to find those? It sounds like looking for a needle in a haystack.
It does. But we have powerful tools now. It relies heavily on bioinformatics and computational immunology. Researchers use sophisticated algorithms to scan the entire protein library of the virus.
The viral proteome.
Right, and compare it against the human proteome, looking for identical or highly similar short peptide sequences. But it's not just about finding a match.
Okay.
The second crucial step is figuring out if those shared sequences can actually bind strongly to our MHC molecules,
Yeah.
the major histocompatibility complex molecules that present antigens to T cells.
Especially the MHC types, the HLA alleles that are already known to be linked to autoimmune diseases.
Precisely, because that's how the immune system sees the mimic.
And you mentioned this binding has to be strong. We're not talking about just any interaction.
No, absolutely not. And this is a really critical point for our physician audience to appreciate the level of detail involved. The binding strength or affinity is measured. It's quantified using something called the IC50, the half maximal inhibitory concentration.
Okay.
To even be considered a potential risk, a T cell epitope, the researchers set a really strict threshold. The predicted IC50 value for both a viral peptide and the matching human peptide had to be less than 250 nanomolars.
250 nm. That sounds incredibly tight. Why such a high bar?
Because affinity dictates activation strength. Think about it. If the binding is weak, maybe 500 nanoram or more, a T cell might recognize it briefly, but the signal probably won't be strong enough to push past all the safety checks, the regulatory mechanisms that maintain self-tolerance.
Especially in places like the thymus, where self-reactive T cells are normally eliminated?
Exactly. But a high affinity binder, something under that 250 nLM mark, gets presented really effectively by the MHC molecule. It delivers a strong, sustained signal to the T cell, one that's much more likely to override those tolerance checkpoints and trigger a full-blown immune response. It separates the real molecular culprits from just background noise.
So it's about ensuring the signal is strong enough to actually cause trouble.
Precisely. True molecular causation.
Okay, so applying that strict filter, what specific human proteins emerged as key targets for SARS-CoV-2 mimicry?
One family of proteins really stood out, the heat shock protein 90 family, HSP90.
HSP90, those are involved in protein folding, right? Basic cellular housekeeping.
Exactly. They're essential chaperone proteins. They help other proteins fold correctly and maintain stability, especially when cells are under stress. Researchers found shared peptide sequences between the SARS-CoV-2 nuclear protein, part of its core structure, and human HSP90 beta, specifically HSP90AB1.
Okay, one specific match.
But it got more compelling. They also found cross-reactive T-cell epitopes, meeting that strict binding criteria between a different SARS-CoV-2 protein, ORF6, and multiple members of the HSP90 family. Not just HSP90AB1, but also HSP90AB3P, HSP90AA1, and HSP90AB2P.
Wow. So multiple hits against this really fundamental protein family. Attacking HSP90 sounds like it would cause chaos pretty much everywhere in the body.
That's the critical insight. HSP90s aren't tissue-specific, they're foundational. They're crucial for the stress response in nearly every cell type. So if the immune system launches a cross-reactive attack against HSP90s, the damage isn't going to be neatly localized, it's going to be systemic.
Which could explain that wide range of post-COVID symptoms.
It provides a strong mechanistic link. In fact, this specific mimicry involving HSP90 is proposed as a potential driver behind conditions like Guillain-Barré syndrome following COVID-19.
Where the immune system attacks peripheral nerves.
Right. GBS often follows infections, and this provides a specific molecular explanation for how SARS-CoV-2 could trigger it by targeting HSP90, which is also present in nerve support cells.
And you mentioned GBS, but were there other neurological connections found?
Yes, there were suggestions of mimicry between SARS-CoV-2 peptides and sequences found in respiratory pacemaker neurons in the brainstem.
Respiratory pacemaker neurons. That could potentially explain some of the breathing difficulties or dysregulation seen in long COVID, couldn't it?
It's a plausible hypothesis. It points towards a potential direct immune attack on central respiratory control systems, going beyond just peripheral nerve issues.
Okay, so this whole concept of molecular mimicry hinges on identifying these high-affinity shared sequences, like with HSP90. But as you said earlier, it only really matters if the person is genetically set up to present that specific mimic to their immune system. It comes back to their HLA type.
It absolutely does. The HLA genes are the blueprint for the MHC molecules. Your specific set of HLA alleles determines which peptides your cells can present effectively. This genetic predisposition is the cornerstone of susceptibility.
Right.
And the source material really emphasizes this. It mentions the development of a tool called the Molecular Mimicry Map, or 3M. It's a sophisticated online application.
What does it do?
It allows researchers to basically check potential cross-reactive sequences, maybe from a new virus, or even a peptide being considered for a vaccine against the whole database of known autoimmune-associated HLA alleles.
So you could plug in a potential vaccine sequence and see if it contains a known mimic that binds strongly to an HLA type linked with, say, multiple sclerosis or type 1 diabetes.
Exactly that. It allows for prospective risk assessment, not just looking back after problems arise.
That feels like a major shift.
It is. And the conclusion drawn from this kind of high-level bioinformatics work is pretty stark. It states explicitly that HLA genetic typing and really investigating individual immune responses are not just academic curiosities anymore.
They're essential.
They're essential steps, necessary if we want to adequately address safety concerns, predict who might not respond to a vaccine, or, crucially, who might be at risk for autoimmune side effects. The hepatitis B vaccine is cited as a long-standing example where these concerns have been debated.
So for our clinical audience, this is suggesting a move towards personalized immunogenetics. Risk stratification based on HLA type might need to become part of safety planning, not just an afterthought based on population statistics.
It's a recognition that immune responses are fundamentally individual. If we know someone has an HLA type that strongly presents a peptide shared between, say, a vaccine component and a critical self-protein like HSP90, well, their individual risk calculation changes dramatically.
It moves beyond population averages to individual predictability.
Yes, managing risk at the individual level.
Okay, so molecular mimicry sets the target. The immune system mistakenly identifies a self-protein because it looks like a pathogen component, especially in someone with the wrong HLA type. Now, let's talk about the effector arm, IL-6, STAT3, and TH17 cells. How do these actually drive the inflammation and damage?
Right. If mimicry points the gun, this axis pulls the trigger and keeps firing. The IL-6 STAT3 TH17 pathway provides the machinery for that sustained chronic tissue-damaging inflammation that defines autoimmune and neuroinflammatory diseases.
Where does it start? We need that initial spark, that danger signal you mentioned.
Correct. It begins with the innate immune system sensing trouble. When a virus or bacterium invades, it displays certain molecular patterns, pathogens-associated molecular patterns, or PMPs, things like viral RNA or bacterial cell wall components.
Okay.
Our cells have sensors for these PMPs called pattern recognition receptors, PRRs. Toll-like receptors, TLRs, are a major family. TLRs 2, 3, 4, 7, 8, 9, they all recognize different microbial signatures.
And that recognition sounds the alarm.
It does. It triggers the rapid production and release of inflammatory cytokines to recruit immune cells. And interleukin-6, IL-6, is one of the first and most prominent ones produced in response to these danger signals.
Now, in really severe situations, like the cytokine storm we saw in severe COVID-19, all roads seem to lead to STAT3 activation. Why is STAT3 so central?
STAT3's signal transducer and activator of transcription 3, yes, it really is a linchpin. In those severe inflammatory states, you get this massive self-amplifying loop. STAT3 activation drives the production of more pro-inflammatory signals, including TNF-alpha and, crucially, IL-6 itself.
So STAT3 tells the cells to make more IL-6.
Exactly. Macrophages, other immune cells, they get caught in this loop. STAT3 directly activates the IL-6 gene transcription. So IL-6 triggers STAT3, and STAT3 triggers more IL-6. It's a positive feedback cycle that just spins out of control, amplifying the inflammation exponentially. That's the cytokine storm.
And the downstream effects of activating STAT3, they seem to explain a lot of the systemic problems seen in patients, right? Like clotting and fibrosis.
Precisely. STAT3 activation turns on a whole host of genes. You see C-reactive proteins, CRP levels, shoot up, something we measure clinically all the time.
Right.
More worryingly, it ramps up production of PAI1, plasminogen activator inhibitor 1. For clinicians, PAI1 is a potent procoagulant. So that STAT3 activation directly links the cytokine storm to the increased risk of blood clots, DVTs, pulmonary emboli, strokes that we saw in severe COVID, and also in other situations like CAR T-cell therapy toxicity.
Okay, so STAT3 links inflammation to thrombosis. What about long-term damage like fibrosis?
That's linked too. STAT3 activation also increases levels of TGF-beta, transforming growth factor beta. TGF-beta is the primary driver of fibrosis, scar tissue formation. This helps explain why patients recovering from severe lung inflammation are at high risk for developing chronic pulmonary fibrosis.
So STAT3 is coordinating the immediate storm, the clotting risk, and the potential for long-term scarring.
It's a central pathogenic mediator, orchestrating multiple damaging effects.
All right, let's connect this back to the adaptive immune system, specifically the T cells. How does IL-6, driven by STAT3, push naive T cells to become those harmful TH17 cells?
This is where the molecular control gets really elegant in a destructive sort of way. The IL-6 signal, acting through STAT3, is the key trigger for TH17 differentiation. And it doesn't just use the standard signaling pathways. It employs a more sophisticated non-canonical route.
How so?
It works by actively shutting down another important gene, a transcription factor called RFX1.
Shutting it down, how does IL-6 do that?
It involves epigenetics. When IL-6 activates STAT3, the phosphorylated STAT3 protein physically travels into the nucleus and binds to a specific region within the RFX1 gene, actually, within intron 7.
Binding inside the gene itself.
Yes, and this binding event recruits enzymes that change the local chromatin structure. It causes histone deacetylation, basically tightening the packaging of the DNA, and increases DNA methylation in that specific regulatory area.
So it's like the cell takes that RFX1 gene, wraps it up tightly, and puts a chemical lock on it.
That's a great analogy. That's functionally what happens. By silencing RFX1 expression, the cell's internal balance shifts dramatically, making it much more likely to differentiate down the TH17 pathway. And IL-6 also provides the signal needed to turn on RR-gamma T.
The master switch for TH17 cells.
Exactly. R-gamma T is the master transcription factor that locks in the TH17 identity and function. So IL-6 does both. It removes a brake, RFX1, and pushes the accelerator, RR-gamma T.
Okay, let me recap the chain. Mimicry provides the target antigen. Innate immunity senses danger, PMPs, and produces IL-6. IL-6 activates STAT3. STAT3 creates a feedback loop for more IL-6, drives clotting and fibrosis pathways, and epigenetically silences RFX1 while boosting ROR-gamma.
Leading to
the generation of pathogenic TH17 cells that attack the mimicked self-tissue.
You've got it. That's the core pathway.
And this clearly explains why TH17 cells are implicated in so many chronic autoimmune diseases.
Absolutely. They are key drivers in rheumatoid arthritis, systemic sclerosis, psoriatic arthritis, conditions where chronic inflammation just chews away at tissues over time.
Does this TH17 link hold up even in acute diseases, like that severe COVID example?
It does. Studies showed clear TH17 activation played a role in COVID-19 severity. It even led to suggestions that blocking the TH17 pathway, maybe targeting IL-17 itself, could be a useful therapeutic strategy.
Interesting.
And there's evidence suggesting that in human chronic autoimmune conditions, the ongoing TH17 inflammation might be even more dependent on IL-6 than what's seen in some mouse models. This makes blocking IL-6 signaling, perhaps using IL-6 receptor antibodies, a very promising strategy. It's already showing promise in things like autoimmune myocarditis.
And we see this kind of TH17 imbalance elsewhere too, right? Like in lupus?
Yes, absolutely. In patients with systemic lupus erythematosus SLE, disease activity often correlates with a shift in the balance between pro-inflammatory TH17 cells and protective regulatory T cells, TREGs. There are too many TH17s and not enough TREGs.
Can that be fixed?
Interestingly, some lab studies showed that you could partially restore that balance, in vitro at least, by using drugs that inhibit autophagy, like chloroquine. It suggests a link between cell metabolism, autophagy, and the TH17 TREG balance.
Wow, okay. That's complex, but paints a clear picture of IL-6 and TH17 as the engine of chronic inflammation. Now, the critical connection. How do external factors, specifically medical interventions like vaccines and their adjuvants, potentially trigger this whole cascade?
Right. This is where things get, well, potentially controversial, but immunologically crucial. We need to connect the internal mechanisms mimicry, IL-6, TH17, to those external triggers. And adjuvants are a key piece of this puzzle.
Adjuvants, the components added to vaccines to make the immune response stronger.
Exactly. And the need for a strong inflammatory trigger, an adjuvant, to actually induce autoimmunity isn't just theoretical. It's how we create most animal models of these diseases in the lab.
Like the main model for multiple sclerosis?
Yes, experimental autoimmune encephalomyelitis, EAE. It's the workhorse model for studying MS. How do you induce EAE? You inject mice, usually genetically susceptible strains, with a myelin protein, a self-antigen from the nervous system.
But the protein alone isn't enough, right?
Correct. Injecting the myelin protein by itself usually leads to tolerance or no response. To get the autoimmune disease, you must inject it along with a powerful adjuvant, typically something called complete Frunz adjuvant, CFA, which contains heat-killed mycobacteria.
Why the adjuvant?
The adjuvant provides those PAMPs, those danger signals we talked about. It forces the antigen-presenting cells to get activated in a highly inflammatory way. This ensures they push the T-cell response towards a pathogenic TH1, or relevant here, TH17 phenotype against the myelin, overriding the normal tolerance mechanisms. No adjuvant, generally no disease.
So the adjuvant essentially provides the kick needed to break tolerance to the self-antigen.
Precisely. It provides the inflammatory context. And the EAE model also shows us it's complex during the disease. New types of antigens, like glycosylated ones, can appear, and antibodies against those can then influence the disease course. It's an ongoing cycle.
Okay, that's the lab model. What about in humans? Are there accepted links between vaccines, possibly via adjuvants and autoimmune events?
There are some well-established, though rare, causal links recognized by medical authorities.
Yep.
For instance, the evidence supports a causal relationship between certain tetanus toxoid-containing vaccines, DT, tetanus toxoid alone, and Guillain-Barré syndrome.
GBS.
Yes, and also anaphylaxis. These are very uncommon events, but the causal link is accepted, and they can rarely be serious. Similarly, there's an established causal link between the MMR vaccine and thrombocytopenia, low platelets, and anaphylaxis.
So, recognized, rare causal links exist. Now, let's address the more contentious area, the critical viewpoints about certain vaccines like hepatitis B and potential links to autoimmunity that tie back into that genetic predisposition idea.
Right. We need to present these viewpoints objectively. There have been persistent concerns raised by critics, citing ongoing reports again, often statistically rare in the whole population, suggesting the hepatitis B vaccine might, in some individuals, trigger serious long-term issues.
What kinds of issues?
The reports mention GBS again, but also central nervous system demyelinating diseases like transverse myelitis, optic neuritis, even potentially multiple sclerosis, and also systemic immune problems like chronic arthritis.
Is there any biological reason to think this could happen, or is it just based on temporal reports?
Well, there is some biologic plausibility argued. First, GBS is known to occur sometimes after a natural hepatitis B infection.
Ah, so the pathogen itself can trigger it.
Correct.
Yep.
Which suggests that components of the virus, or the immune response to them, can be cross-reactive in susceptible people.
Ah.
So the argument follows that vaccine components derived from the virus could theoretically carry a similar, though likely much, much lower risk. Demyelinating events in general have also been reported after various vaccinations, not just Hep B.
Okay. And the source material pointed out something really crucial about who tends to report these types of reactions to the Hep B vaccine, connecting it back to genetics.
Yes, this is a key analytical point. The observation highlighted is that a significant proportion of the patients reporting these serious adverse reactions are of Caucasian background.
Why is that significant?
Because it aligns perfectly with our understanding of HLA genetics. Specific HLA alleles, which are strongly associated with susceptibility to various autoimmune diseases, have different frequencies in different ethnic populations. Caucasians have a different profile of common HLA alleles compared to, say, East Asian or Polynesian populations.
So if the people reporting reactions are mostly Caucasian, it might imply that specific HLA alleles common in that group are involved in the susceptibility.
That's the strong implication, and it raises a critical question about how we assess vaccine safety based on large population studies. Well, the source material points out that if the major safety studies establishing the vaccine's overall good profile were conducted in populations with very different ethnic makeups, the examples given were Taiwan, Alaska, New Zealand, then those studies might not have had enough statistical power to detect a rare adverse event that primarily occurs in individuals with specific Caucasian HLA types.
Meaning a risk that's statistically tiny for the whole world might be significantly higher, though still rare overall, within a specific genetically defined subgroup.
Exactly. It highlights the potential inadequacy of a purely population-based safety assessment for predicting risk in genetically distinct subgroups.
This really forces the issue of personalized risk assessment, doesn't it? It suggests that a population-level safety statistic might not fully capture the risk for an individual with a known high-risk HLA profile.
It absolutely does. It shifts the focus from broad statistics back to individual immunogenetics. If you know a patient has an HLA allele strongly associated with presenting a mimic peptide found in the vaccine, their personal risk profile is different, even if the overall population risk is extremely low.
Which circles back to the call for potentially incorporating HLA typing into risk assessment.
It reinforces that argument, yes, moving towards managing risk based on individual genetic makeup.
Okay. We've traced the mimicry, the IL-6 TH17 inflammation, and the potential role of triggers like adjuvants, especially in genetically susceptible individuals. Now, let's pull this thread all the way through to neurodevelopment. How does this inflammatory cascade potentially connect to autism spectrum disorder?
The evidence linking immune dysregulation to ASD pathophysiology is really quite strong now. And it's not just about behavior. we see actual physical differences in the brains of children with ASD or those who later develop it.
You mean structural changes?
Yes, significant, measurable structural changes. One of the most consistent findings is in the cerebellum studies show a noticeable decrease in the number of Purkinje cells.
Those are large neurons critical for motor control, coordination, even cognitive functions.
Exactly. Losing them points to significant disruption. And even earlier in development, neuroimaging studies have picked up differences. For instance, a pattern of hyperexpansion of the brain's cortical surface area between 6 and 12 months of age.
So the surface of the brain grows unusually fast early on.
Yes, and this seems to precede the later finding of overall brain volume overgrowth seen between 12 and 24 months in some children with ASD. These early structural changes strongly suggest something is interfering with normal brain development pathways.
And inflammation is a prime suspect.
A very prime suspect,
Uh,
especially inflammation originating during pregnancy.
maternal infection during pregnancy.
Yes. There's a growing body of research suggesting that maternal infections, whether viral, bacterial, even fungal, can act as triggers. The idea is that the infection sets up a state of chronic inflammation in the mother, which then adversely affects the developing fetus.
How strong is that link?
One large analysis found that any reported maternal infection during pregnancy increased the odds of the child later developing ASD by about 12%. It's a statistically significant association.
What's the proposed mechanism? Is the fetus getting infected directly, or is it something else?
Both mechanisms are possible, but the one getting the most attention, especially in the context of inflammation, is the indirect effect. It's called maternal immune activation, or MIA.
MIA, so it's not the bug itself, but the mother's response to the bug that causes the problem.
That's the prevailing hypothesis. The idea is that the mother mounts a strong inflammatory response to her infection, producing lots of cytokines like IL-6, activating pathways like TH17, the very things we've been discussing. These maternal inflammatory molecules can then cross the placenta. Some might even cross the developing fetal blood-brain barrier.
And directly interfere with brain development.
Exactly. They arrive at a critical time when neurons are migrating, forming connections, synapses are being pruned, and these inflammatory signals can disrupt all those delicate processes, especially if the fetus itself has genetic susceptibilities. So the mother's acute infection translates into a potentially chronic neuroinflammatory state and altered developmental trajectory for the child.
That provides a powerful inflammatory bridge connecting systemic immunity to neurodevelopment. What about the microbiome? That comes up a lot in discussions about ASD and immunity.
It does, and for good reason. Patients with ASD consistently show signs of gut dysbiosis and imbalance in their gut microbial communities.
What kind of imbalance?
The most common finding is an altered ratio between the two major bacterial phyla, Firmicutes and Bacteroidetes. Typically in ASD, you see lower levels of Firmicutes and significantly higher levels of Bacteroidetes.
And this isn't just a random difference, it has functional consequences.
The evidence suggests it does. In rodent models, for example, artificially increasing the levels of Bacteroidetes has been shown to correlate strongly with the emergence of autistic-like behaviors.
How might gut bacteria influence brain function and inflammation?
There are several proposed routes. One is through changes in the production of short-chain fatty acids, SCFAs, like butyrate. These are metabolites produced by gut bacteria that are crucial for maintaining the integrity of the gut lining and also for modulating immune cell function, including T-cell responses systemically.
So the wrong bacteria mean the wrong signals for the immune system.
Potentially, yes. Another idea is that certain bacteria, perhaps those overabundant Bacteroidetes, might produce metabolites or even peptide mimics that constantly stimulate low-grade inflammation. This inflammation could signal to the brain via the vagus nerve, or inflammatory molecules could leak into the bloodstream from a more permeable gut barrier, leaky gut.
Perpetuating that neuroimmune activation cycle.
Precisely. It sets up a scenario where the gut microbiome is potentially contributing to, or at least exacerbating, the chronic low-grade inflammation that impacts neurodevelopment in susceptible individuals. The gut and brain are in constant communication, and in dysbiosis, that communication might be predominantly inflammatory.
It really highlights how interconnected everything is: genetics, environment, infection, immunity, microbiome, and brain development.
It's incredibly interconnected.
Okay, let's try to synthesize this. We've covered a lot of ground, from predicting molecular mimics using bioinformatics to the downstream effects of IL-6 and TH17, the role of adjuvants, and the potential links to ASD via maternal immune activation and dysbiosis.
I think the synthesis really comes down to this.
Hmm.
We can now see plausible, specific molecular pathways connecting external triggers infections, potentially adjuvants to complex outcomes like autoimmunity or neurodevelopmental disruption.
It's not just vague associations anymore.
No, we have molecular mimicry providing the specific antigenic target, but it only becomes dangerous in the context of the right, or perhaps wrong, HLA genetics,
Then the trigger activates innate immunity, leading to that IL-6 surge.
which activates STAT3, creating the inflammatory feedback loop and, crucially, epigenetically programming T cells toward that destructive TH17 phenotype.
And that chronic TH17 inflammation then drives the pathology, whether it's attacking joints, nerves, or potentially interfering with brain development.
Exactly. And the recognition that specific HLA alleles are key determinants of who is susceptible to this cascade, especially in response to things like infections or potentially vaccine components, that really shifts the paradigm.
It moves us towards predictive
It
immunogenetics.
does. We're moving beyond just population statistics.
Which brings us to a really challenging point for clinical practice today. We seem to have the knowledge, the tools are developing, the mechanisms are becoming clearer, but implementing this kind of personalized risk assessment based on immunogenetics is still, well, not standard practice at all.
It's minimal outside of very specialized areas, like transplantation.
So that leads us to our final provocative thought for you, our audience of physicians and researchers, to really think about.
Given everything we've discussed, the growing evidence for molecular mimicry, the critical role of HLA genetics in determining susceptibility to severe outcomes after infection or potentially rare adverse events after vaccination, how quickly do we need to adapt?
How fast does clinical practice need to evolve? From the way we even design vaccines and adjuvants in the first place, right through to how we counsel individual patients and document potential risks.
Do we need to start routinely incorporating personalized immunogenetic testing, like HLA typing?
If we can potentially predict, based on someone's HLA profile, that they are at significantly higher risk for developing a severe chronic TH17-mediated autoimmune reaction to a specific trigger, is it still okay to rely only on general population safety data when advising them?
Does risk stratification using genetics move from being a nice-to-have research idea to becoming an essential, maybe even obligatory, part of preventative medicine and informed consent?
It raises huge questions about standardization, cost, access, interpretation. But if the goal is truly personalized in preventative medicine, avoiding predictable harm in susceptible individuals, maybe these are hurdles we need to tackle head on right now.
It feels like the next frontier for safety and precision in immunology and medicine generally. A complex challenge, no doubt.
Indeed. Something to seriously consider. Thank you for joining us for this deep dive today.